Yeah, so one cell goes through the first half of meiosis to produce 2, and then each one of those goes through the second half of meiosis to produce, each one of these produces 2 more. So you go from 1 to 4 at the end, and this is done. Each one of these has half the DNA. DNA. You have a whole lot more than 4. But the process for meiosis starts with 1 and ends with 4. The mitosis starts with 1 and ends with 2. It's a significant difference. And any of you who are biologically female, this process is done in you. You are not still doing this. It's over. They do, except theirs is different. Like, perhaps this wasn't discussed in grade 9. It absolutely should have been. Any of you who are biologically female were born with all of the eggs you are ever going to have. You've got them all. Your body is not producing more. So, like, part of developing as an embryo, female embryo, is your body will do all of its meiosis at this stage, producing all of the eggs that you will ever have. That's why menopause exists. For those of you who have mothers of that age, they might talk to you about the fact that they're going through menopause. The reason that menopause happens is because the eggs are done. And the body, as a result, the hormone changes in a female's body, woman's body, cause menopause. Really hoping this wasn't brand new news to all of you. In biological males, that is not the case. Biological males' bodies will undergo meiosis constantly from inside the womb until death. No, when they were in the womb. When they were in the womb. Before my son was born, but developed as an embryo, his body was producing sperm cells. And his body is constantly producing them until he dies. A biological male's body always undergoes meiosis. Eggs have a long shelf life. Sperm have none. 0 shelf life. So meiosis it's important. If it doesn't happen, sexual reproduction doesn't happen. Birds, mammals, fish don't exist without this process. One and done in women, like forever in biological males. Yeah, some do. 3, yeah, I learned this last night. While I was reading before bed. There are 3 species that go through menopause. Humans are 1. The other, and it's called the grandmother theory. So there's, technically speaking, there are only 3 species on our planet that have what we would consider to be grandmothers, where they are done mothering and have an additional role to play. Those 3 species are humans, elephants, and killer whales, orcas. Those are the only species that we know of on our planet whose elder females go through menopause, which is really interesting from an evolutionary biology perspective. It's really quite interesting because that means that there's a role to, so in other species, when a female is unable to reproduce, they die. Their life has served its purpose. But in these 3 species, humans, orcas, and elephants, there's a role yet to play for females who can no longer reproduce. And that's really interesting to discuss at that philosophical level. It's really quite interesting. Yeah, it's a good question. I read it last night. Literally, I'm not making that up. What else? So bringing it back to grade 9, mitosis, meiosis. Mitosis is a form of sexual reproduction, but there are several types of sexual reproduction that you would have looked at. Does anyone remember what kinds of asexual reproduction types you studied? There weren't too many. There was like 7. It was a lot, but maybe as a class we can remember what they are. Was it even 7? Might have been less than that. Might have only been 6. Well, I'll tell you one of them. Mitosis as a form of asexual reproduction is called binary fission. Hopefully that name triggers some stuff. Kohi? That's number 7. Cloning in the lab. Is that what you had to present on for me? Yeah. If any one of you who had me, you had a presentation to do on this. You should remember your own type at least. Brooklyn. Spores, yes. Spores are down here. You betcha. Anyone who had me, what was your presentation topic? You don't remember? Shame. I'm sealing. It's a hard unit for a lot of people. There's lots to memorize. Binary fission, spores, fragmentation, love it. Goes up here at the top. Starfish undergo fragmentation. If you chop up a starfish, as long as each piece has access to the central disc, a part of the central disc, you will grow a new starfish. That is a genetic clone of the original. It is. It's terrifying because starfish are actually pretty barbaric. They will wipe out whole ecosystems if there's too many of them. Yes, the potatoes. There was, the potatoes are the one with the runners. Even I don't remember. Well, let's call it runners. I don't remember if that's what it's called, but that's what we're going to call it. Yeah, the roots go out. Yeah. There's grafting is another form of vegetative reproduction. The vegetables, all the plants will have their own. Vegetative reproduction. So runners is one. It's the one, strawberries are the big example of runners. They're the ones that they send out runners along the ground, and then every so often out of those runners, roots will go down into the dirt, and you get a new strawberry plant at that location. Potatoes do something similar, except they're under the ground when they do it. The last one I have here is budding. No, these three, the top 3 are all animal species. Do this. And plant species do this. Plants and fungus do this. Propagation. That is the word, yes. Propagation. I didn't even get to teach this unit last year. It's been a long time since I taught the bio unit. This was Mr. Gerbrandt who did this for you. Yes, he did. Well, a chunk of chemistry. I finished chemistry. But yes. Oh, no, he didn't do chemistry. He did biology and physics. I did chemistry and astronomy. He started the first week. Yeah, he started the first week or 2. Okay, so this is, basically this page is grade 9. That's all of it. The details, there are more details, but this is basically grade 9. So let's move on to grade 10, now that we've reviewed. Grade 10 starts with a discussion on DNA. DNA you've learned before. So what can you tell me about DNA? All living things that we see are closely linked to something we cannot see. Some of you have seen this. Hands up if you did the strawberry DNA lab in grade 9 or in middle school. It gets done to death. I don't do it in grade 10 because almost all of you have done it. You take the strawberry, you crush it up, you add something to it, you filter it out, you get rid of all the strawberry bits, and you're left with something that looks like snot. And your science teacher will be like, oh, that snot is actually DNA. And that's it. It's a weird looking substance. And it's not like you can actually see the bits of the DNA that make up the DNA, but you believe your science teacher who tells you it's DNA. She tells you it's DNA, must be DNA. What else could it be, right? So we have this molecule within us, all of us. DNA is wildly controversial. I mean, you might hear it say that you share certain percent of DNA with this other organism. That's true. But when DNA is as long as it is, you might share 99% of your DNA with a chimp. But that 1% of DNA is still a lot of DNA. And 1% difference makes a significant difference. You can speak. You have thumbs. You're not covered in hair from head to toe, where monkeys and chimps are. Gorillas' arms are longer than their legs. Yours aren't. Like, that 1% difference is significant. You have 77% DNA shared with a tree. But that 23% difference is significant. So all life on our planet has this molecule DNA, and this DNA codes for different things. What does DNA stand for? Who remembers? Yes. Deoxy. Ribonucleic. There's the D in DNA. There's the N in DNA. So the deoxyribo is the D, the nucleic is the N, and A is the acid. Deoxyribonucleic acid is a genetic material that stores information. It codes for you. Sorry? Allegedly. I'm not the biologist. I'm a physics chemistry guy. I'm just going with what the biologists have told me. It's called deoxyribonucleic acid. It is not, I wouldn't classify it as an acid the way chemists define an acid, but that is what this molecule is called, deoxyribonucleic acid. Genetic material that stores information, codes for you. But it doesn't just, it's more complicated than that, because you can have genetically identical twins that look different, that sound different, that think different. I've known them. I have godchildren. Like, I am their godfather. Not like the movie The Godfather, but like, if their parents die, my wife and I get guardianship. I have godchildren that are genetically identical twins. And I can assure you, they do not look identical. So there's something more to who you are than just this. So I don't want to oversimplify DNA, but it does code for you. It is what makes you you. DNA is responsible for variation. It's not the only thing responsible for variation, clearly, because you can get genetically identical twins that aren't genetically identical. And it's even more interesting if you have genetically identical twins that marry genetically identical twins and then have kids. Because technically speaking, kids of siblings should be cousins. But biologically, they're actually siblings. They're genetic siblings, even though they are children of... Do you follow you picturing what I'm saying? Okay, I'll draw it for those of you who are struggling. I'll draw it. Genetics is weird. It's fascinating, but it's weird. So you have twin identical twin dudes. They're going to go ahead and they're going to marry. This is my horribly stereotypic stick person. Don't take offense to my stereotypical stick person. Identical twins. Genetically identical twins. These two get married. Those two get married. They have kids of their own. The kids that are produced from this marriage and the kids that are produced from this marriage, however many there are, in the normal world, when these aren't identical twins, we call these cousins, yeah? These are siblings, together they are cousins. But when you have genetically identical twins and genetically identical twins, these are genetic siblings, not cousins.
That's so cool.
Genetically speaking, if these two are identical, it doesn't actually matter. From a genetic perspective, it doesn't matter which one's which. But obviously, there's a relationship that exists there. These are different people with their own likes and dislikes. But as far as the genetics is concerned, it doesn't matter which is weird and interesting. Yes, Evelyn. Not on the genetic level. If they're identical twins, yeah, it's weird. It's weird. So DNA is responsible for variation among living things. There are other words for variation here. I'm going to give you one of them. When we talk about variation within a species, we're talking about diversity. Is the extra word I'm going to toss in there. Most cells of an organism contain genetic information that has an influence on its appearance and life processes. There are cells on you that don't. Does anyone know which cells do not contain a copy of your DNA? There are cells that you have that do not contain a copy of your DNA. There are cells that you have that do not contain a copy of your DNA. Most of your cells do. Red blood cells are one of them. Red blood cells do not contain a copy of your DNA. No, white blood cells do. This is good. In the physics unit and the chemistry unit, I've got a lot of answers to a lot of whys. Biology, I don't. I'm just going to be straight with you. I don't. There's a lot of whys you're going to ask me in biology where I don't have a good answer for you. I want you to ask them, but do be aware, I will be saying I don't know a lot more this unit. Chemistry and physics, I got it. Biology is a little weird. It's cool, but it's a little weird. Okay, so red blood cells are one. They do not contain a copy of your DNA. White blood cells do. Red blood cells are, well, there is a reason for that. I'll give you my overall understanding of the reason once we got some more of these cells down. Hair cells do not. Because they don't. I'll explain it in a sec. I know why. This one I can answer, but I'm not going to tell you yet. There is a portion of your hair that does. But there is a portion of your hair that does not. Does anyone want to lend me a piece of your hair? Someone can pull out their hair from the base. This is one single piece. All I need is a single piece. You want a piece? I can tell you right now. It'll work or not. This is you? Which one is attached to your head? It's not that. That one is. I know. This one won't work. I know what those are. I know it's. That's what I was going to say. I'm impressed. No, that one's not going to work either. Oh, is that going to be like the white piece? Yeah. You need that piece. You want that piece? No. Yeah, you need the follicle. Any of you who ever, if I had a pair of tweezers, I can get you in the room. My beard hairs are all short enough that they all do, but I can't. Well, maybe I can. So I can get one out. But they're coarse. No, I don't get any follicles. So here's a piece of Brooklyn's hair. And I think at this point you all understand what I mean. Hey, what's up? Oh, you don't need to take Evelyn. She's great. Yeah, so for those of you who have no idea what I'm talking about, can you even see this on the screen? Yeah, you totally can. There's Brooklyn's hair. And this is the part that was attached to her head. You got the white part? Yeah, sweet. I'm gonna love it. So we're gonna compare Brooklyn's hair with Brooklyn's hair. Okay. So we got different pieces of hair. We got this one. Where are you? I don't know. We're gonna find it. We'll get there. Where's the hair? And you're blonde. Yours is gonna be even harder to see. Oh, you know what? Beggars can't be choosers. You're doing great. Okay, let's see if I can see here. Where is he here? Let's do that, and then zoom it in. I want to focus it. Oh man, this is going to be hard to see. Where's the word smaller? Can we see that hair above there? Okay, right on the end of that, can you see the little ball that's there on the end? That's called the follicle. That is the only portion of hair that contains DNA. All the rest of all of this does not. There is no DNA there. If there's, then hair would be useless. Yes, and then Brooklyn, your darker, thicker hair is way easier to see. This one does not. I don't want that end. I want this end. So then here's the end of Brooklyn's hair. No follicle. So this particular piece of hair, if we wanted to figure out who it belonged to, we couldn't. If we wanted to figure out the other one, it would point directly to you. So if I get murdered at my desk later, the only DNA here is going to be mine and hers. Okay, so red blood cells, hair cells, your fingernail or your nails, fingers and toes. Nail cells do not include DNA. And believe it or not, skin cells don't either. Fingerprint and DNA is different. Your fingerprint, there's a particular pattern to it, but it's not connected to DNA. And the reason why at least some of these don't include, that aren't your DNA, is because they are just straight protein. So you perhaps have an understanding of what a protein is. A protein is a section of DNA, not the whole thing. Hair cells and nail cells, red blood cells, are a protein chunk. So it's a portion of your DNA, but not all of it. And skin cells, that's just a fun fact. I have no idea why, but they don't. Cells contain two types of DNA. Two types of nucleic acids. One of them is DNA. Does anyone know the other name? RNA. Good. RNA. Right there, called ribonucleic acid. It's missing the deoxy part. And nucleic acids are large molecules made up of smaller components. Again, they're hard to see, but they're long. They're very long molecules. They're made up of smaller components called, does anyone know what the name of the smaller component is? Of both DNA and RNA. Nucleic acids, both of these are large molecules made up of small pieces. Actually, it's actually on the page already. I'll just give you the name. It's called a nucleotide. Yeah, I know, it's on the page. There are these nucleotide structures linked together to form your full DNA chain. This figure down here, this guy, is a nucleotide, this bottom left. The figure below, that guy, shows the general structure of nucleotides. There's a phosphate group at the end of it, there's a sugar group in the middle, and then there's a nitrogenous base. And so that's what we're going to put in here. There's a phosphate group. And a phosphate group is just a term for a compound made up of the polyatomic ion phosphate, PO4, and something else. There's a phosphate group at the end of every nucleotide. There's a sugar. It's not sugar the way you typically think of sugar. When I say the word sugar, you probably think of what? Table sugar, which is what's the chemical name of table sugar? Starts with a G. Glucose. Table sugar is glucose. In DNA, this sugar is when we were naming organic compounds, I told you that all sugars' names end with the same thing. Does anyone remember what it was? I mean, if you think about glucose, if they all end the same way, glucose, sucrose, they all end in ose. So, where is it? This guy here. Deoxyribose is the name of the sugar. Deoxyribose is the sugar that makes up your DNA. And it contains a nitrogen-containing molecule called a nitrogenous base. This should be review. I'm hoping this is review. Nope, not review. It's been a long time. Sorry? This part? Okay. Does anyone remember what the nitrogenous bases are called? There are 4 of them. We code them as A, C, G, and T. Has anyone seen these 4 things before? Where? Science 9? In science, you talked about them in Science 9? Who's your teacher for Science 9? Ms. Gill, okay. So there are 4 of them. Do we remember? Yeah, Haley. Adenine is 1. Did you look it up? Or you?
It's on the page.
Oh, it's on the page. That's right. Adenine. I'm hoping you remember. Cytosine, guanine. Sometimes I can keep your attention long enough for students to realize not to realize it's not there, but it's there. And thiamine. Those are the nitrogenous bases. Some students have heard of this before. Some students have not. Sometimes it gets taught in grade 9. Sometimes it comes up in middle school. DNA gets taught awkwardly at different stages throughout your guys' educational career. I don't know. Their guess is as good as mine. When you're ready to flip the page, flip the page. This I almost certainly guarantee you have not seen before. So those nitrogenous bases, adenine, guanine, thiamine, and cytosine, have different structures to them, and their structures dictate how they link up and behave. Adenine and guanine both have 2 rings as a part of their structure. They are called purines. And thymine and cytosine, for whatever reason, only have 1. And they are called pyrimidines. I am not going to expect you to remember that. Purines and pyrimidines. At this point, I'm just trying to expose you to as much of the language as I can. So for those of you who are like, yeah, biology, and you're going to go off and that's going to be your field of study of choice, I want you to at least have been exposed to some of that language. I'm not going to, a multiple choice question is not going to show up on a test. What are the names of the rings on the, no, I don't care about that. Yes. If you flip back to the very front page, look here, look in this middle section, Evelyn. There's a section on the front, on the title page called nitrogenous bases. It's got adenine, guanine, thymine, and cytosine, and it's got the Lewis diagrams of each. This is what it looks like as a 3D structure. This is what the Lewis diagram would look like. Yes? Here? What O's are you talking about? Those are circle, like here, these are just circle, that means there's, oh yeah, those are oxygens. Yeah. N is nitrogen, H is hydrogen, O is oxygen. And the part where they link up, the thing that isn't labeled, is carbon. And that's in a simplified molecule diagram. Those junctions that are not labeled are all carbons. Just to simplify it, that's how they roll. So these are the structures. So you can see here, adenine and guanine have 2 rings, ring there, ring there. And thymine and cytosine each only have 1. It's just the structure that those molecules hold. Why? I don't know if there's a person alive on the planet that could tell you the answer to that question. That's just how the structure of the chemicals is, or the compounds have shown up. I'm reasonably confident there's not a person alive who could tell you why. But the implications of that are significant. So DNA is a molecule made up of 2 strands of nucleotides, which is going to be important when we talk about the function of DNA. The structure of DNA is made of 2 strands of nucleotides linked together. So it's got kind of this coil appearance to it, which you guys are going to make for me today, in a way. It all coils together, loops into a chromosome. But when you uncoil it, it looks like this ladder structure. There's a lot going on in that diagram. The 2 strands are joined by hydrogen bonds. Remember those bonds I talked to you about yesterday? Or the reason why the breaking of those was the reason why ice water is colder than the ice cubes themselves? These hydrogen bonds link the DNA together. Yes, Evelyn? They're not all. So these bars here are these nucleotides. So every single one of these bars is made up of one of these and one of these that link together. So the adenine and guanine, they each have 2 rings. Thymine and cytosine each only have 1 ring. And this thing is every one of these rungs is 3 rings long. So these 2, we're going to talk about it a little bit more in a second, but you're asking the questions, I want to answer it now. Every one of these rings is either made up of an adenine-thymine togetherness group or a guanine-cytosine group. The order from rung to rung is the code that makes you, yes. But every one of these rungs is made up of a AT bond or a CG bond. Every one of those rungs, and so because of this, the ring structure limits what kind of combinations you can have. So the structure of DNA looks like a twisted ladder, which you're going to see hopefully today. We'll see if we get there. Or another name for that is called a double helix. And the discovery of this structure was a big deal. Now, Nobel Prize winning big deal. It was. The guys who came up with the structure for DNA, they won the Nobel Prize in biology. The sides of the ladder are made up of the sugar phosphate groups, and the sugar phosphate, we call that the backbone. So that's this, in this picture up here, that's this thicker part. This is the sugar phosphate part. The rungs are the nucleotide part. When you're ready, flip the page. There you go, Brooklyn. This key will get you in. Go to the second one. Or you can go to the women's one, it's up to you. Okay, so what I tried to describe to you a second ago, Evelyn, is this structure here. So you can see these rungs here are made up of either the adenine thymine group or the guanine cytosine group. And then you got the sugar phosphate backbone. The rungs are the nucleotide part, yes. That's due to the fact that there are as many as there are. When there are so many, we're talking, I don't even want to guess the number. I'm going to figure out the number. Rungs. I love the questions. The questions are great. Oh boy. Okay, so there is no firm number, but when we're talking about a combination of multiple billions of these things, the odds of having those codes be identical in unrelated people is impossible is not the right word. The odds are more than that. Like it's just astronomically small. So much so that it's effectively impossible. There are only 8 billion people alive now. Estimates say that there might only be 100 billion people who have ever lived, beginning to now, and lived and died and are on the planet right now. Maybe something like 100 billion, 113 billion, I think is the largest number I've ever seen as an estimate. And when there are more than that possible combinations, that right there should be enough, I think, to tell you why. Each rung of the ladder is made up of the 2 nitrogenous bases, as I've said. In the image, I think Brooklyn, you need the keys too? No, she forgot. Oh, gotcha. In the image on the right, you can see each set of bases of H base pairs. These dotted lines in the middle here are the hydrogen bonds I was referring to before. So these guys right here, the hydrogen bonds are right here. And it's really important that these are hydrogen bonds and not ionic or covalent bonds. If they were ionic and covalent, when they got pulled apart, it would shatter the structure of the molecules themselves. Like the molecule wouldn't be the molecule anymore. But because they're hydrogen bonds, and those are bonds that exist between a compound and another compound, when you separate them, it doesn't destroy the structure. And so your DNA, we're gonna look at how your DNA does this, but your DNA unzips and rezips along this hydrogen bond line every time it needs to be duplicated. So every time meiosis or mitosis happen, or anytime your DNA needs to make another protein, needs to make more hair, okay, cool, unzip the DNA. Needs to make hemoglobin, the protein responsible for helping your red blood cells carry oxygen. Unzip the DNA. And every time it unzips, it can rezip without destroying its structure because of these hydrogen bonds. Brooklyn's hand was up before Evelyn's.
So you said that like the dot parts like make hair, right?
Yeah.
So if you have like cancer, are they like technically unzipped? Like for
You're asking why people go bald when they have chemo?
yeah, like when they're able to unzip because like not is that like something different?
That's different.
Changing part of the DNA structure at that point. Because if they don't often
No, it just kills your follicle. Kills your follicles.
So it has nothing to do with like the hydrogen bond.
No, chemo's a poison. Well, any medication in a large enough dose is a poison, but chemo's particularly bad. But it's necessary. Evelyn.
I feel it's similar. It's like as people get older, does this slow down? Yeah. Because like people stop like.
There are things. Yes. So the short answer is it doesn't slow down, but it doesn't happen as effectively. So there's a piece of a DNA we're not going to talk about. There's a piece of DNA that we're not going to talk about called a telomere. Telomere? Pretty sure it's called a telomere. I might be mispronouncing it, but that's what it's called. And every time DNA gets replicated, the telomeres get shorter and shorter and shorter and shorter. And when they get too short, the DNA that particular section of DNA can no longer be replicated. And so when that starts to happen, that's when mistakes can start to creep in more aggressively. And that's when aging starts to dominate. You can see the effects of aging. The effects of aging are partially caused by the shortening of these telomeres. Your body's not doing as good a job as it was of replenishing you. It's more complicated than that, but that's the explanation I can give you at this stage. You cannot change the fact that those telomeres are going to shorten. Your DNA has a limit to how many times it can be copied. And when that limit is reached, it's not getting copied anymore. And when your DNA is not getting copied anymore over enough of you, you can connect the dots. You die. I mean, there's other pieces to things that cause us to live and die. It's not all genetic, but there is a genetic upper limit to the human race. You can't take care of your DNA. There's a genetic upper limit to how long a human being can live. I mean, and every old person will tell you what their secret to life is. There's one old guy in Britain, he was interviewed, he's like, yeah, I'm 105. Bourbon every day. Yeah. I assure you, bourbon every day has resulted in the death of other people. So, I mean, why some people live longer than others, I don't know. Some of it no. Some of it, yeah. Okay, so in the image to the right, here, you can see that the base rungs of the ladder must be paired a specific way. G goes with C, A goes with T, adenine will always pair with thymine. Guanine always pairs with cytosine, they just do. And nitrogenous bases that bond or pair together in this way are called complementary bases. Is the language that goes here. So, if I opened up a strand of DNA and I told you that the one side of the DNA was coded like this, A, T, G, C, G, T, T, A, C, if I told you that that's what the left side looked like, the right side would have to look like what? A always bonds with T, C always bonds with G. What would the right side of this DNA chain have to look like? So yeah, so what would that be? T, A, C, G, C, A, T, G. That is the only way that those could code. These would only go together this way. In someone, somewhere, I'm sure that exists. I have no idea what that is. I just made that up. But absolutely, this would code for something somewhere in someone. You bet. Why not? The problem here being, now in a human being, you might have a chain like this, except it's not 1, 2, 3, 4, 5, 6, 7, 8, 9 long. It might be 30 billion long. So if you have a 30 billion long chain of letters, yeah, I'm sure ATGCGTTAC exists. Do it, man. Sorry? How this codes? No, you were. No. The human genome is the same length in every human. Every typical human has the same length code. No, I'm not even going to say average. Typical. There are people who have extra chromosomes, for example. There's a whole syndrome called Down syndrome because there's an extra chromosome. Extra chromosome means extra huge chunk of DNA. Yep. No. It's way more complicated than that. We will talk about Down syndrome a little bit. Okay, so we're gonna dang it. We're gonna start something today that we're gonna have to finish tomorrow. You guys have so many good questions. Okay, here we're gonna start with this. Take one, pass them all the way back. 1, 2, 3, 4. Take and pass. 1, 2, 3. Elomere? Elomere. 6 of those. Okay, so all y'all have exactly the same copy of a string of DNA, which is going to make this, not totally a true exercise, but we're going to go for it. First thing you're going to do is I want you to cut out the rectangle, like the whole outer shape, along the outside line. 1, 2, 3. Okay, everyone I just gave scissors to, those scissors go back in this bag. Everyone that I'm about to give scissors to, your scissors come back to me. Everyone who I just gave scissors to goes back in this bag over here. Everyone who I'm going to give scissors to comes back to me. We need scissors. Yes, you do. Cut around the outside edge. All the way around. All the way around the outside edge. Hang on to it. Oh, don't fold it that way. Don't fold it that way. You're going to fold it this way. We're doing origami, guys. Oh. You're going to do origami. You have to fold it a very particular way. Do origami. You can keep it straight. You know what? You do not know what origami is. You're going to all do origami. You're all going to make a tiny piece of an origami DNA chain, and we're going to put all the chains together and make one real big chain. Let's go. Well, we're going to finish tomorrow. Okay. Once you've cut it up, I want you to give me the scissors back. Return the scissors. See you later. Good luck. See you tomorrow. Good luck. And you can put your paper scraps into the recycling bin. Return your scissors. Paper scraps to the recycling bin. And I don't know what the easiest way to do this is. I'm going to try and talk you through what you need to do. No wonder I look. Oh, there we go. I got it. You got it. So we're going to all try and do this together. Okay. Please don't. Okay, so we're gonna try and all do this together. We'll get as far as we get before the day is done. You've got yours. Here is an example. You're gonna take this thing and you're gonna fold it in half lengthwise, making all the creases as firm as possible. So you can see here, this, what you see here goes on the outside. This thick black line in the middle is what you're trying to fold along, like this. Fold it really quite carefully on that line, and you want the crease to be tight. Like once you folded it, lay it down on your desk and run your fingernail up and down it. You want that to be a hard crease. You are committing to a fold like you've never done origami before, like you've never committed to a fold in your life. Those of you who have commitment issues, I'm sorry. You're committing to a fold in a big way. There will be no unfolding this. Don't screw up. Like so many of your other commitments in life, don't screw those up. Number two, you're going to hold the paper so that the thick lines are diagonal and the thin lines are horizontal. So those are like, you can see here, one side has the thick line diagonal and one has the thick line horizontal. Hold it so that those thick lines are diagonal and the thin lines look like this. You want to see this side. Okay, that's what's facing you. And fold the top segment down onto the next one down, like this. Just the top one, you're folding it on that thin line, and then unfold. So you've got your thing here, you're going to fold it down like this, and then unfold. You can demonstrate with the manuals if you like. So on manuals, I'm holding it so that the thick lines are diagonal. So you're holding it like this, and then you're going to fold it along the thin line, and then you're going to unfold. Oh yeah. So that's the first one, then you unfold. So you said something looks like this. You've unfolded. And now you're going to fold the top two segments down. Basically, you folded one down, you're going to unfold it. You're going to fold two down, you're going to fold it. You're going to fold three down. So all the way down, it's all folding the same way. You're folding all those guys down along the thin horizontal line. Now I folded this one and undone. Now you're going to fold the next one. Let me know if you need a hand, or if you need specific, my hand's on. Sorry. Okay, so you do the first one. The thick lines are diagonal. So you go back. First one. And the second one, you're going to fold it on that line there. Now you've got this fold. This fold. Anyone else in the hand? Yeah, keep going all the way down. Yeah. Repeat for all the segments. You're folding it down on every segment all the way down, on every one of those thin lines. Yeah, well, keep going. Yeah, that's right. Yeah, keep going. If you want the thick lines diagonal, so this is the side you want to look at. So refold those. You can fold them the other way. Yeah. Remember, you want the thick lines. You want to be looking at the side where the thicks are diagonal.
Inside.